Good day, students. Today we are going to talk about the cytoskeleton of the cell. In previous lectures, we have talked about the single membrane bound organelles, double membrane bound organelles, and non-membrane bound organelles, as well as cytoplasm, cell wall, cell membrane, and the nucleus. So, in brief, we can say we have studied the overall structure of a cell, and we have also studied the differences between the plant cell and the animal cell. Now we are also going to today's lecture we are going to discuss one more important structure that is known as cytoskeleton. So first of all, how we will define the cytoskeleton? Actually, cytoskeleton is nothing but a framework of filamentous structures. So we can say that cytoskeleton is extensive, flexible, supporting and dynamic framework of the filamentous structures. And this filamentous structure or this framework is suspended in the cytoplasm or the cytosol. Now let me explain what these terms, that is extensive, flexible, supporting and dynamic framework. Extensive because the cytoskeleton is present throughout the cell in the cytoplasm. It remains suspended in the cytoplasm, so it is the extensive framework. Flexible because it is highly flexible, and it is supporting because it supports the structure of the cell, and dynamic framework because it is continuously forming and then breaking down into the constituents. So it is continuously undergoing change, therefore it is called as dynamic, and it is the framework which supports the cell. Now, the cytoskeleton is composed of three types of protein filaments. And these three types are microfilaments, macrofilaments, or rather we can call them microtubules, and lastly the intermediate filaments. These three types of the filaments which form the cytoskeleton are distinguished on the basis of their thickness and the type of protein they are made up of. For example, in case of microfilaments, its thickness is least or minimum, that is approximately 8 nanometer in diameter, whereas microtubules can be called as the thickest filaments because they have a diameter of approximately 25 nanometers, whereas the intermediate filaments are in between the two, that is microfilaments and microtubules, that is they have diameter in between that of microfilament and the microtubules. So they have the diameter of approximately 8 to 10 nanometers. We'll see the differences in more detail shortly. So let us discuss the basic differences between these three types of the filaments. In the first column we will write the characteristic features of the microfilaments. In the middle one we will write about the microtubules. And the last one will be having the characteristic features of intermediate filaments. The first difference in between the three is on the basis of their thickness, as I have said you earlier also told you. The first difference is the thickness. So we can say that microfilaments have the least diameter, or they are the thinnest filaments, with the approximate diameter of 8 nanometer. You must be knowing the scale of nano. Nano means 10 raised to power 9 meters. In case of microtubules, they are thicker in comparison to microfilament and hollow. So we can say they are thick and hollow filament. With the maximum diameter, in the case of all the three, they have the diameter of approximately 25 nanometers. The intermediate filaments, as the name suggests, they are intermediate in between in the thickness, that is the thickness of microfilaments and microtubules. So on an average we can say they have approximately 8 to 10 nanometer of diameter. Now the second difference is on the basis of the constituent proteins. In case of microfilaments, they are composed mainly of actin. Besides actin, the microfilaments also contain small amounts of myosin, tropomyosin, troponin, actinin, etc. And these are similar to the muscles protein. Whereas the microtubules are composed of tubulin protein, whereas the intermediate filaments are heterogeneous in nature because they are composed of different types of proteins in different cells. So they cannot be said to be composed mainly of one or two proteins, as in case of microfilaments and microtubules. We can here say they are heterogeneous. Hetero means different. Now coming to their functions, on the basis of function also, these three types of filaments differ. Firstly, if we talk about the function of microfilaments, their, their main function is to maintain cell shape and provide the mechanical support. Besides this, they are also responsible for the motility or providing motility to the cell. If we talk about the functions of microtubules, they are very important filaments because they also help in maintaining the cell shape. Besides microfilaments, their first function can be written as that they maintain cell shape along with the microfilaments. Now besides maintaining cell shape, the microtubules are also involved in the intracellular transport of the macromolecules. That means the transport of molecules between different parts of the cell is carried out with the help of the microtubules. So we can say they help in or involved in intracellular transport of the macromolecules. Besides this, the microtubules are also the part of microtubular organelles like cilia, flagella, as well as their basal bodies, besides their centrioles and division spindles. All these organelles are very important for a cell. For example, cilia and flagella along with their basal bodies help in the movement of the cell, and the centrioles and division spindles are very important during the time of cell division. The division spindles can be also called as the spindle fiber. Now coming to the functions of the intermediate filaments, these are also very important since they form a cage-like network around the nucleus, and therefore it connects the nucleus to the plasma membrane. We can say it forms cage-like network around the nucleus, and this network connects the nucleus to the plasma membrane. Besides this, the intermediate filaments are also present at the cell-cell junction. It is the point where the two cells are associated or attached. So, it is present at the cell-cell junction also. As I told you that the intermediate filaments are heterogeneous in nature in terms of their protein composition because they are composed of different types of proteins in different cells, and one such example can be the keratin protein of skin. This keratin of skin is found in the epidermal cells. So in epidermal cells, the intermediate filaments are made up of protein keratin. One example of the intermediate filaments is the keratin of the skin epidermal cell. So this is all about the cytoskeleton of the cell, which provides the supporting framework. Now we'll discuss the basic difference between the two types of cell, that is prokaryotic cell and the eukaryotic cell. So we can say that on the basis of the organization, the cells are of two types, that is the prokaryotic cells and the eukaryotic cells. So we are going to discuss the types of cell on the basis of type of organization. As I told you that there are two types of cell, and these two types are eukaryotic and prokaryotic. We will discuss these two types of cells and differences between them in detail. But the basic difference between these two types of cell is that in eukaryotic cells they have true nucleus. U means true and karyon means nucleus. So a eukaryotic cell has true nucleus. By now you must be familiar with the structure of nucleus and the function it plays. Whereas in case of prokaryotes, the pro word means primitive and karyon means nucleus. So they have primitive type of nucleus, which is better called as the nucleoid. Now let us see the differences between these. What are the other differences between these two types of the cells? As I told you that the basic difference between these two types of the cell is on the basis of the presence of nucleus, whether it is the true type of nucleus or primitive nucleus. So we can say in case of nucleus it is primitive types, or it is also known as incipient nucleus. Incipient means primitive type of nucleus. And this primitive type of nucleus is known as the nucleoid. This primitive or incipient nucleus, which is also known as nucleoid, lacks nuclear membrane. It also lacks the nucleolus. So we can say that the nuclear material, the genetic material, which is the DNA, it is present as a single closed loop without any histone proteins. Generally, in the eukaryotic cells, what happens is that the DNA is present in the form of chromosomes, and in these chromosomes, the DNA is associated with some basic proteins which are called as histones. So we can say in case of prokaryotic cell, DNA is present as a single closed loop without histone protein. This DNA can be also called as the chromosome. So we can say in the prokaryotic cell only one chromosome is present, that too in the form of a closed circular loop. And in this chromosome DNA is not associated with the basic histone proteins. Now coming to the eukaryotic cells. In case of eukaryotic cell, they have true nucleus, and we all know what are the characteristic features of a nucleus, that it has a nuclear membrane, that is the nucleus is surrounded by a covering membrane that is called as nuclear membrane. Also, the nucleus contains nucleolus. Now coming to the genetic material, that is DNA. The DNA exists in the form of multiple structures which are called as chromosomes, and in these chromosomes the DNA is associated with the basic proteins which are known as histones. Now this is the basic difference between the eukaryotic cell and prokaryotic cell. What are the other differences? Let us discuss them. The second difference can be cited in the terms of the respiratory system. In case of the prokaryotic cells, these well-defined respiratory organelles are absent. Instead, in case of prokaryotic cells, the respiratory system is associated with the plasma membrane. So we can say mitochondria, which is the organelle responsible for respiration, is absent, and these plasma membrane structures which are having the respiratory system are called as the mesosomes. And this respiratory system associated with plasma membrane is termed as mesosome. Besides this mesosome, no other organelle is found like mitochondria. So we can say that mitochondria is absent. Whereas in the case of the eukaryotic cells, we all know that they have their respiratory system associated with the mitochondria. We have studied the structure of mitochondria in detail. So we can say that mitochondria is present in the eukaryotic cell. Now coming to the terms of photosynthetic apparatus. If the bacteria is if the prokaryotic cell is photosynthetic, the photosynthetic apparatus is associated with the internal membrane, that is plasma membrane. Besides plasma membrane, it contains the system of membranes which lie in the cytoplasm, and these internal membranes are responsible for the photosynthesis process. So we can say photosynthesis, the photosynthetic apparatus is associated with the internal membrane. So we can say that the photosynthetic organelle, that is chloroplast, is absent. Chloroplast is a type of plastid which contains green pigment chlorophyll. But in the case of eukaryotic cells, they have their, if they are photosynthetic like the plant cells, they have their photosynthetic apparatus present in the chloroplast. Now coming to the other organelles, besides these, the prokaryotic cells lack the membrane-bound organelles like Golgi bodies, endoplasmic reticulum, lysosomes, and vacuoles. We have talked earlier about the mitochondria and chloroplast. They are also absent in the prokaryotic cells. Whereas in case of eukaryotic cells, the membrane-bound organelles are present. Till now we have discussed that the membrane-bound organelles are absent, but the membrane without membrane organelles, that is membrane-less organelles like ribosomes, are present in the prokaryotes also. But the prokaryotes contain different type of ribosome, that is known as 70S ribosome. Whereas the eukaryotic cells have 80S type of ribosome. We have discussed what the S stands for in the structure of ribosome. The S stands for the Seeberg unit that represent the sedimentation coefficient. So eukaryotic cells contain ATS type of ribosome. If we talk about the cell wall, the eukaryotic cells have cell wall only in the plant cells, whereas the prokaryotic cells, they generally have cell wall. See, all the types of prokaryotic cells generally have cell wall. But in case of eukaryotic cells, the cell wall is present only in the plant cell. Besides plant cell, the fungal cells also contain the cell wall. There is one more structure outside the cell wall. It is the smooth and slimy structure called as the capsule. The capsule may be or may not be present in the case of prokaryotic cells, but it is never present in the case of eukaryotic cells. So we can say capsule may be present in the prokaryotic cell, and if it is present, it will be present always outside the cell wall. So it is a slimy capsule is a slimy smooth structure which is present outside the cell wall in the case of prokaryotic cells if it is present. It might be absent also, but if we talk about the eukaryotic cells, they never have any capsule like structure. Now coming to the last difference, how the cell divide. If we talk about the prokaryotic cells, they generally divide by simple methods like binary fission, whereas the eukaryotic cells divide by the mitosis. So we can say the process of cell division, which is known as mitosis, is not found in the prokaryotic cell, whereas if we talk about the eukaryotic cell, they can divide by the process of mitosis. So we say mitosis do occur in the eukaryotic cell. So this is all about the differences between the two types of cell, that is the prokaryotic cells and the eukaryotic cells. In the next lecture we will be discussing about some of the important points regarding the cell.